The next few slides really are background. We've got some real experts in the field in this room today, so apologies, but I think it is worth just describing this so everybody understands where we're coming from and what's within scope and what's not within scope. So these next slides really bring everybody up to the same speed of what we're describing here as the objectives. We're saying that many different classes of therapeutic drugs cause drug-induced liver injury in the human population. We're very familiar from the literature with the fact that people have classically identified reproducible type A dose-dependent toxicity of the type we see with paracetamol as one end of the spectrum. And typically, this is something that's readily reproduced in various animal species as well as in man. And it's a type of toxicity we will pick up during routine preclinical safety testing. This is within scope for our prediction, because although this is something we would pick up using during conventional preclinical safety testing, we're looking for the 3 R's replacement refinement. We don't want to put a vast amount of time and effort into discovering a molecule that turns out to be hepatotoxic when we undertake regulatory safety studies. We want to deselect those molecules in the course of drug discovery. And there are no fully characterized ways to do that currently, so that is definitely within scope. We've also got fully within scope the other pattern of drug-induced liver injury. Many people think of this as a continuum, but classically in the textbooks it's described as another pattern of liver injury. This is the drugs that don't cause overt dose-dependent liver injury in man or in preclinical species, and typically they sail through preclinical safety assessment studies and don't exhibit any evidence of anything concerning at all. But we get to largest human populations in clinical trials or even post-licensing, and we start to see the odd individual, maybe 1 in 100, maybe 1 in 1,000, maybe 1 in 10,000 individuals, develops an abnormal response and manifests in potentially severe drug-induced liver injury. You know, everybody's worst nightmare, the idiosyncratic pattern of liver injury. This is fully within scope for the call. And we believe this distinguishes our call from any other attempt at predicting this area in the past. We are saying we need to tackle propensity to cause idiosyncratic drug-induced liver injury. We believe that idiosyncratic drug-induced liver injury is a complex end product of multiple factors. Some of the factors would be drug-specific because there are some drugs that have a significant possibility of causing idiosyncratic liver injury. And some of the problem of susceptibility is clearly patient-specific. This is a preclinical call. We are not going after patient-specific factors. We're not going after genetics, for example. We are going after drug-specific factors. So I'll pick an example of a drug series. If we were developing PPAR active agents at the moment, and we had triglytazone, rosiglitazone, and pioglitazone as candidate drugs, we would obviously not want to take forward triglytazone. We would be more than happy to take forward pioglitazone as our preferred candidate. And actually, if we got rosiglitazone or pioglitazone, we'd actually do okay. Unfortunately, rosiglitazone is now being withdrawn because of cardiovascular toxicity, but it's not because of liver toxicity. So we need to have approaches to get after these drug-specific factors, and I could pick other classes of drugs and make the same kind of associations. There are clearly some bad actor molecules that have a much higher propensity to cause idiosyncratic drug-induced liver injury than others. And these drug-specific factors are what we're going after within this call. This is things about incidence, an example of it. Although idiosyncratic drug-induced liver injury caused by individual drugs is infrequent, the problem is that many hundreds of drugs which are on the market can cause idiosyncratic drug-induced liver injury, and many of them are extremely widely marketed drugs. So if you aggregate together the vast number of patients exposed to these agents and add together the vast number of drugs that can cause these problems, although it's an infrequent problem with individual drugs, it actually becomes a major socio-economic problem when you aggregate it all together. That's why it's so important that we tackle it. And back to the issue about why it's so important, and particularly from a patient health perspective, we need to have ways to tackle risk of idiosyncratic toxicity, because we don't want to be licensing new agents that are going to make a susceptible patient sick. If you're the susceptible patient and you get life-threatening liver injury, it doesn't matter that it's rare, you're affected. And back in my academic career, I once upon a time did a lot of work on the anaesthetic halothane. And in 1 year, I saw 7 patients in acute liver failure caused by halothane. A very rare adverse reaction. I know that I'm resistant because I've inhaled enough halothane over the years that I know I'm non-susceptible. But if you're a susceptible individual, it's really horrible and traumatic. We're looking to reduce the likelihood that those sort of rare, unpleasant effects will happen with new drugs that are coming through drug development and getting through to the human population. Yeah, final thing about technologies and capabilities. And the scientific community has been coming up with mechanisms and has been coming up with promising experimental model systems. So I believe personally that propensity to cause idiosyncratic toxicity could be tractable if we're smart enough and we pick the right kind of model systems and do the right sort of studies. And I think we're in a place where we've got a possibility to do that. Expected impact on the R&D process? Well, ultimately we're going to have a new series of approaches which, if successful, will reduce unpleasant consequences in our clinical trials. We know that toxicity observed in clinical trials is actually a major reason why drugs are falling out of clinical development. Kinetics isn't a major problem anymore. Efficacy can be compromised, but actually the big one that's dangling above the parapet and isn't being tackled is toxicity. And one of the most frequent types of toxicity we see in preclinical species and in man is drug-induced liver injury. So we believe if we're successful, we'll have improved preclinical prediction of drug-induced liver injury and actually more successful pharmaceutical companies. If we can manage that within the timescale of this project, I'll be amazed. I think we'll have some approaches that will be a step change in how we approach things within pharmaceutical companies. And the big beneficiary will be the drug industry after this program completes in about 5 years' time. And then ultimately, 5 or 10 years down the track from that, as this translates into new pharmaceutical agents reaching the marketplace, we ultimately have a massive benefit for the human population who are now getting good drugs through to market that have managed to make their way through the drug development process and have not fallen over because of unpredicted toxicities. So ultimately, farmers will be a major benefit, and a bit further down the track, everybody in the European population will be as well. We've got many good ideas that we think could translate themselves into good drugs if only we could have safe drugs. And increasingly, we require safe drugs. We don't want to take risks in the human population. So that's our thinking of this. I'll be very happy to field any questions you have, and my co-conspirator in this, Richard in the corner, will join me in responding to any questions you may have. Thanks for your attention.